Welcome everybody to the April edition of BioStock Live from a crisp spring day in Stockholm, Sweden. I'm extremely happy to say that today we have a little bit extended program. We have 8 interesting companies who are going to join us today. And we have showcases, but we also have panel discussions. And we are actually going to start right away with an, I'm hoping, exciting panel discussion concerning the Alzheimer's disease area. And we have invited today the 3 major players in the Swedish biotech scene who are in the Alzheimer area. And I am very pleased to welcome from AlzeCure, Johan Sandin, CEO. And from BioArctic, we have Gunilla Oswald, CEO. And then Alzinova, Per Wester, CEO of Alzinova. And the special guest today is also Mattias Häggblom from the portfolio manager of Robur Medica, the Swedish, is the largest healthcare fund in Sweden?
One of the larger, at least, yeah.
One of the larger, yeah. You're all very welcome. So, Alzheimer's disease, it's a huge challenge, both in terms for the patient and their relatives and close ones, but also for the healthcare business and society in general. Why is Alzheimer's disease such a huge challenge? If we start with you, Gunilla.
So I think Alzheimer's disease is a disease that most of us are familiar with. I think most of us know someone with Alzheimer's disease, and we have relatives and so forth. And it's really a disease that affects also relatives and neighbors and has a huge impact, as you said, on the society. Today, there are only symptomatic treatments on the market that can help to relieve some of the symptoms. What is lacking completely today is disease-modifying treatments that is meant to affect the underlying disease. And also, we need better diagnostic tools to be able to identify the patients at an early stage. Because the earlier we can give the treatment, the better effect the patients can have for a disease-modifying treatment. So I think there is a huge need for better treatments for this population. And I'm really happy to be here today and talk more about that. And I think that we should be very proud of being Swedish people, because BioArctic, we have one of the most advanced programs where there is most hope in the Alzheimer's disease arena. And I'll talk more about the positive phase 2b results. And we are soon now in phase 3. And we're doing everything we can to get the trial to Sweden.
Thank you very much. And it says over 50%, even actually 65% of the population will at some time after 65 years of age be impacted by dementia or Alzheimer's disease. And could you say, is this number actually increasing, Johan?
So with regard to the number of people being affected, so if you look at the age of 65, approximately a few percent has Alzheimer's disease, and then that doubles every 5 years approximately. So this is clearly a huge health problem, and the major risk factor for getting dementias and Alzheimer's disease is high age. So with increasing life expectancy, you will actually see an increase in the number of people getting the disease. And actually, if you look at from a global perspective, the highest increases we are currently seeing is in the low and middle income countries, where the quality and the available capital for people making them live longer and actually getting the disease. So we're seeing an increase primarily in the low and middle income countries, but also obviously in Europe and the States.
And with the prevalence increasing and the huge challenge of not having any disease-modifying treatments available, Per, what do you say? What's your approach to trying to change that?
Well, the brain is always complicated, and I think that of all the diseases that humans are saving nowadays, we know that they are making much steps forward in cancer, and also progress is happening within cardiovascular diseases. But the brain is different, and the reason for that becoming so complicated, I think, is because there is no really good animal models that you can test your candidates on before you go into humans, because the human brain is so advanced, and we see so many specific things happening to a brain which is deteriorating. And this is, I think, one of the biggest problems that we have, because it's not really possible to test the drugs until you are actually giving them to humans. I think it's very much value to, if possible, when you do the first clinical trial in humans, you should go for patients directly, because then you can also try to catch one of these biomarkers which is also connected to the disease. And that's then the first time the drug is really tested. It's not really tested when it's in preclinical settings. So I think that is one of the reasons why it has been so difficult, and it has been so I mean, some people say that you take out a transgenic mouse and you find a drug which can actually treat this mouse to become normal. This doesn't mean that we have found a new treatment for Alzheimer's disease for humans. So,
unfortunately,
no.
Historically we've seen, many failures in this area. I think I saw a summary that said that it was 85 projects or something like that in the last decade that had been terminated because of different reasons. Gunilla, you said that BioArctic, you are one of the companies that are come the furthest, at least here in Sweden. What's
globally, I would say, but thanks to ASI, our partner.
Okay. And what do you think is going to be the clue that makes the breakthrough?
No, I think, I just want also to continue to build on what Per just said. I've been working with Alzheimer's disease for drug industry for many years, for AstraZeneca for many years, and for BioArctic for 6 years. And everyone should know that drug development comes with risk, and of course Alzheimer's disease is also comes with risk. Different ways to mitigate the risk is to see if you can have a strong link to genetic aspects, and if you can have a strong link and show histopathology in combination with the best we can do from preclinical trials. So what I think is really important is to understand as much as possible about your mechanism of action of the compound. And BioArctic, we are founded by Professor Lars Landfelt from Uppsala University. And he found first the Swedish mutation and then the Arctic mutation of Alzheimer's disease. And then he saw that these patients with the Arctic mutation, they had increased levels of a misfolded toxic soluble aggregated form of A-beta that he called protofibrils or oligomers. And I think it is important to selectively target those toxic species. And that is what BAN2401 is doing, and that is the compound which has shown the most promising effect in clinical stage globally, and it's driven by our partner ASI. And ASI is the compound who developed donepezil or Aricept, which is the most commonly sold symptomatic treatment. So it's a company who really focus on Alzheimer's disease in a broad way. So I really think the mechanism of action, and we think selectively binding those toxic misfolded species, not the monomers and not so much the fibrils, but really those toxic. And it seems like the whole field is going more and more there. We have seen that solanezumab, which went mainly for the monomers, it didn't work. It had a small effect, but not enough. We just recently saw that aducanumab was stopped, and that's mainly targeting the fibrils. So we think BAN2401 really is targeting the right species, which is called the protofibrous or oligomer. And I think it's so cool. It's a Swedish invention. I'm really proud of that. And it's really ASI who should have a lot of compliments for how they are driving the clinical program.
And speaking of programs, Johan, Alzecure, you have a twofold approach, so to speak. Can you tell us something about that?
Yes, so we're having actually two different type of therapies targeting then two cardinal features of the disease. One is the, as Gunilla mentioned, the buildup of amyloid aggregates in the brain, and the other one is then focused on the neuronal dysfunction and the loss of communication between nerve cells that cause the cognitive deficits in the disease. So targeting two different kind of patient populations, if you will. One, NeuroRestore, which is then a symptomatic therapy targeting patients in the later stages of the disease, and Alstatin, which is a gamma secretase modulator targeting the production of amyloid and buildup of amyloid aggregates in the brain.
And looking at you mentioned, Gunilla, early diagnostics as one tool. Why would that be? Why is that important? Early diagnostics.
Well, I think mostly because when we see patients get symptoms, many people or scientists now say it's too late. So you need to find these patients before symptoms occur. And that is a very, very difficult target to reach because everybody is not agreeing upon what these diagnostic tools should actually show. I know that in Gothenburg we have Professor Kai Blennow and Henrik Zetterberg, who is among the world leaders in this field, and it's very, very interesting. We talk to them regularly, of course, because we are almost next door to them, and they help us a lot. But they are developing new kinds of assays to detect from CNF and CSF and from also blood samples. And there is an interesting cooperation that we are actually involved in with them to see if it's possible to find the right species from the CSF, sorry, from patients actually. So this is something that is developed also in Sweden, which is again very good. And I think besides that, I mean, of course, coming back to what we talked a little bit about earlier, when we talk about this A-beta target, we all know now that not all A-beta are the same. Some of it is protective and others are toxic. And if you have a treatment which targets something which is not toxic, you might even worsen the situation for the patient. We have seen that a lot with the BACE inhibitors, which were presented during last year, where they actually saw that brains were shrinking. This is not what you want to see if you treat the patient with Alzheimer's. And that's just an example. We have all heard about this ARIAS, which is also a mistargeted side effect. So because the toxic forms are so rare, these protofibrils or oligomers or whatever we call them, they are so rare, So if you should be able to find them, you must have antibodies available only for them. If they can also, each antibody can also bind to other species, it's not probably going to find the oligomers, toxic forms, and you will not see an effect. This is something that is crucial when you go in with the treatment, and that's why we think, with all respect to what has been happening earlier days, that the real oligomer specific treatment has not been tested yet in humans. But of course, some people have other ideas how they can show that their treatments are oligomer specific to 100%, and that is. But whether that is shown or not, I think this is absolutely needed for a treatment to become to give a strong effect. Of course, there can be signs of effects if you have treatments that binds different sources.
Gunilla, I got the feeling that you wanted to reply on that.
Yes. So I agree with you. BAN2401 doesn't target 100%, but it's oligomer or protofibril selective, and it does not bind the monomers, but it binds the fibrils to some extent. So you're right there that it's not 100%, but we have shown clinical effect, and it's not 100% effect. But in the phase 2b trial that was reported last year, it's a huge trial, 856 patients in the US, Canada, Europe, Japan, and South Korea. They were treated for 18 months, either with placebo or with one of the 5 doses of BAN2401. And what I think is really promising, and why I really believe in this, is that we had an effect on clinical outcomes, 3 different clinical scales. We also showed that we are clearing amyloid from the brain. It's not the right amyloid, but it's the best we can show. And also, really importantly, we also show that we have an effect on neurodegenerative markers like total tau, phospho-tau, neurogranin, and neurofilament light. All of those together says that this has the potential to become a disease-modifying treatment. And you're right, if we think, and many with us, that if you're targeting the fibrils, you also have a risk of getting this side effect called area E, amyloid-related imaging abnormalities, where it looks on MRI scans like you have a brain edema, and it's mainly without any symptoms, but it looks, you see it on MRI, but it's all reversible, and we had less than 10% on our top dose who had that. So I think that BAN2401 in this huge trial, it showed good effect on clinical measurements, good effect on clearing amyloid from the brain, good disease-modifying biomarkers, together with a good safety tolerability profile. So Eisai, our partner, they have discussed with authorities, and after these discussions with authorities in the US, Europe, and Japan, they have decided to do 1 phase 3 trial instead of 2. So now they have initiated a huge trial with 1,566 patients globally. That will test the one dose that showed the most promising results versus placebo treatment for 18 months with these earlier Alzheimer's disease patients. So I just want to build on what you said earlier also, that previously, and those compounds on the market today for symptomatic treatments, they have mild, moderate, and severe Alzheimer's disease. We are targeting what we call early Alzheimer's disease. That is mild Alzheimer, and the stage before, called mild cognitive impairment with Alzheimer pathology. Then interestingly, the regulatory authorities in Europe and in the US are also advocating for that we should try to go even earlier, which they call stage 1 and stage 2, which is even before it's called prodromal or mild cognitive impairment. And then it's really important to have the biomarkers to identify the patients and to have sensitive clinical scales to be able to. And Eisai has also announced that they are looking into the possibility and explore BAN2401 in those earlier stages with consortia.
And I'll just like to add and intersect there as well. Just to mention that alstatin, that our disease-modifying treatment affects the production of all amyloid forms, so regardless if it's oligomers, fibrils, protofibrils, or plaques. But just before we leave the diagnostics area, I think I'd like to build on what Gunilla said, because I think this is a really critical area where Sweden has played a quite important role with Kai Blanov's and Henrik Zetterberg's groups, for example. I think where we're now seeing much more focus on novel initiatives we've seen in the US, for example, the Diagnostics Accelerator program that Bill Gates and also now the Amazon CEO has been funding, and I think those are critical initiatives. And we're also seeing the kind of start of other, not only kind of spinal taps and brain scans, but also blood-based biomarkers, which I think also is kind of a segue into moving into this earlier population that Gunilla mentioned. I think clearly the whole field is now seeing, and that's been shown now in many clinical studies, that the disease starts many years before symptom onset. So we know now the disease probably starts some 10 to 15 years before symptom onset. So we need to go into earlier in the disease, and I think as was said previously, both the FDA and EMA are issuing new guidelines to move into early stages of the disease. They have also seen the kind of that we've moved forward in the research around Alzheimer's disease. We know more about the disease now than we did 10 years ago. So we also need to change the regulatory requirements and be able to move in early into the disease.
So then, with a complex situation, Matthias, I know you monitor the international scene very closely. What's your take on the Alzheimer's challenge from an investor's perspective?
I guess one comment would be it's one of the last large diseases with no relevant treatment, so everyone can understand that anyone with a treatment effect will have a commercial success, which is why historically so many companies have invested so heavily in this field. The last 20 years, pharma wrote a report 2018 that $700 billion US has been invested in Alzheimer's, with limited success. 4 drugs the last 20 years, one of them a combination of the previously approved. So the opportunity is large. I'd like to add maybe the perspective also that I sense an increasing debate within the community whether or not the A-beta hypothesis is the relevant. And I do that as I'm not part of the A-beta hypothesis, but I think it's relevant to lift that because it's been the dominant hypothesis for the last 2-3 decades, and we now had 9 phase 3 trials with antibodies failing there. And I think it's difficult to, if you've been a researcher, spent 2-3 decades of your life to finally admit that, maybe this was the wrong path. 30 to 40% of Alzheimer's patients does not have plaque when they die in their disease. So there's still a debate around all what we've been discussing and so forth. But I was surprised when I read the paper in New England Journal of Medicine 2018 from one of the groups who had published their phase 3 data on BACE inhibitors from the Merck, that under discussion said maybe it's time to reconsider if this is truly the right approach. And I think we have an interesting decision to be made by one of the strongest supporters of Alzheimer's research, Biogen, coming up here in how they will move forward with BAN 2401. They are in collaboration with ASI, and they are refusing to comment on what their intention is post their disappointing aducanumab outcome here earlier this year. They saw the stock price drop sharply, and they are currently doing a strategic review what to do since they spent so much money on this. So I think I don't have a strong opinion. I'm just saying that there is a debate ongoing in the community. I find that interesting, and we've heard various alternatives to explain the historical outcomes, but it's still a fact that we have had very limited success, unfortunately, with drug development so far in this field.
You're both waving. You go first, Pat.
I think everybody is hearing what you're saying, but I still think that what most of the specialists say is that there is difference between different A-beta species, and that is very important. It might actually be that we are this close to find a treatment which will work within A-beta, even though there has been a lot of failures. Don't forget that all these failures have shown some signs of effect. Many of them, not all of them, but many of them has shown some signs of effect, which means that there is really something in there. I think that nobody longer believes that the plaque load is going to change. If you release the plaque load, that will then make the patient become better. But there are other forms of A-beta which are much more important, and they are much more difficult to trace. They are so difficult to trace. It's like finding the needle in a haystack. And if you have all these antibodies which targets to everything out there, some of them might by chance bind to one of the oligomeric forms, but most of them are out there doing harm, more or less, because they cause these ARIAs and make things become worse instead of better. So I think it's a big mistake, and I think many of the specialists around the globe also states that it's not A-beta as a field, but it's A-beta oligomeric forms that is the real problem here. And that must be not forgotten, because it's so important that we don't take everything into one, to solve all the things with one solution when there are so many different species.
Can I continue to build on that, please? So I agree completely with Per. I think don't say A-beta and think that it's the same. It's a lot of different species within A-beta. You need to understand, unfortunately. And I think that also what BAN2401 has shown that it targets the oligomers and protofibrils of A-beta, but it also has effect on tau, total tau, phospho-tau, neurogranin, and neurofilament light, neurodegenerative markers, which is more downstream in the process of pathology. So I think, and there is, in all scientific debates, there are, those who believe in amyloid and those who believe in tau and so forth. So there is always debate in scientific community. But I think that let's look at the data and the fact, what we have shown with BAN2401. We have an effect on A-beta, but we also have an effect on tau and other neurodegenerative markers, which I think is important to look at. And then also, I don't think that we will sort everything with the first compound. I'm sure there will be combination therapies between different amyloid species and tau, and also a small molecule that maybe then targets the buildup, and an antibody that also can take away the toxic species even when there is, because there is a combination of buildup and elimination, which makes an imbalance. And an antibody can really also take away those toxic species. And maybe a vaccine as well in the future. So I really think there is a need for many compounds, and I really hope that many companies continue to fight this awful disease. I really, really hope so. And I think that we are close to seeing some true progress here.
Yeah. Well, let's hope so. Johan?
Yeah, just to build on that as well, I also believe strongly in combination therapies. I mean, what we're looking at is a disease which has a progression over 30 years. And it would be, I mean, there are clearly different processes along the disease progression. So, I mean, in the really late stage of the disease, probably there will be different types of therapies than early in the disease. We know, I mean, just looking at the amyloid hypothesis, because I think it's a fair question when these types of trials fail, to discuss and debate in around what type of grounds do we have to believe in this hypothesis? Well, the amyloid hypothesis has quite strong, or have very strong, genetic support. I mean, if we look at familiar forms of Alzheimer's disease, all forms all cases of familial disease are based on mutations that result in accumulation of amyloid and early onset disease. It's also seen in Down syndrome patients where you have this trisomy, which also duplicates then the APP gene, causing then early onset Alzheimer's disease. So I think there's strong genetic evidence, there's clearly strong biomarker evidence, there's clearly strong biological evidence for the hypothesis. But again, to be able to test the hypothesis as we've been discussing here, I think it's about choosing the right target, choosing the right stage of the disease, before you can say anything about whether the hypothesis is wrong or right. So I don't think it's black and white. So clearly, I think there are more things to take into consideration.
So hope lives on. Matthias, from an investor's perspective again, when you have a field like the Alzheimer field, which has been troubled with so many disappointments over the years, and a recent failure, what does that mean for the investors' general attitude towards going into this field? Because the promise is huge if they find something.
Well, happily so, we can see we have 3 companies here today represented, in Sweden. So, there is still interest and support from financial markets to fund these initiatives, which is great. And we're also seeing, increased interest from some of the more, philanthropic players like Bill Gates. 2 years back now has been increasingly interested not only in malaria and HIV, but also Alzheimer's specifically, given the challenge that he and his group sees, the next decades ahead. Unfortunately, we've also seen some large cap pharma companies exiting the field. So we've seen the well-known decision by AstraZeneca to exit the field and open up possibilities for others. But we've also seen some of their other fellows. I think Pfizer departed here a few years ago. Some are more unknown. Is Lilly still committed? Is Merck still committed? They've had large failures. I think they publicly say that they are committed, but if you're looking at their pipelines, they are less so. So the field is somewhat changing. I refer to Biogen coming up here again. They will have to make a decision how committed they are shortly. They're under big pressure now from financial markets. Will they spend more money on this? And at the same time, we're seeing new entrants here. So we have Sanofi signing a big deal last year. We have Novo Nordisk exploring GLP-1 in Alzheimer's. So there's still not, there's not net people going down in terms of interested parties here, but this field is somewhat changing, I'd say.
Mm. And then you can argue for perseverance. You have to keep working focused and take in all the new what you learn and add it to the knowledge bank, so to speak. But on the other hand, there are also completely new approaches. I read something about interesting, speaking of diagnostics, that they were going to use the voice to actually detect if you have an onset of dementia or Alzheimer's, which sounds most like science fiction to me, but hopefully it can help. Yeah. From your perspective, Matthias, what do you think? Where will we be in 5 years?
5 years is a short time in drug development, so unfortunately, but also we have IP to protect those breakthroughs that then happens to really get a return on those initiatives. 5 years, I mean, we're going to have a number of additional phase 3 trials reading out, including getting last year BAN 2401, and we're going to have some additional progress in clinical development, maybe towards at least proof of concept in phase 2. Beyond that, there is also the pharma industry isn't so interested in talking about it, but preventive initiatives is worth highlighting. So there was a publication in Lancet in 2014 by Norton, who concluded that a third of the Alzheimer's population have risk factors that can be addressed and potentially have a helpful benefit. A group at Karolinska, also I think in Lancet in 2015, had a finger study where they had a bit more than 1,000 Finnish patients that were not Alzheimer patients, but beyond 60 and fairly average, slightly below average cognitive scores, and they had if I recall correctly, in a 2-year trial where they used dietary advice, exercise, social interaction, brain examples of using your brain and exercising that way. They had a 25% improvement on a score they used for cognitive improvement. So again, that doesn't fit all, and I guess maybe that's a reminder that there's a lot of initiatives here that can be helpful, and there is not going to be a single one.
I really, really support what you said there, Mattias. I think, everyone should take on as much as you can about the lifestyle, and we should start as early as possible, and then use thinking about how we exercise and how we interact with people and get social stimulants and so forth. And then I think it's a combination of different treatments that we will see. So at BioArctic, we have then BAN2401, which Eisai is driving, and they and Biogen are sharing the costs. And there we will see the results Eisai has said by 2022. So then we know if the phase 3 trial has been able to confirm the phase 2b positive results. And then at BioArctic, we also work on 4 other programs. We have a backup to BAN2401, but then we have 3 early programs, 3 completely different mechanisms of actions, all disease-modifying and all antibodies, but different mechanism of actions. And I think it's really, really important that we continue with the research in this area, because there will not be one thing that fits all. I think it really, really is important with continued discovery to really help to get better treatments for the patients. And I really hope, and I believe that we are on the right track and have the first antibody on the market to help patients. But there will be need for more. So I really, really hope there will be continued effort in this really important area.
Per, you have something?
Yes, I just want to agree with everything you say here, and I also think that it's very much valuable to see that the more specific drugs that are coming to markets now are probably going to be more successful and probably being more effective. Because they are not diluted, so to speak, to the wrong targets, but getting their actual target, which is the one that needs to be found. And don't forget that the oligomeric forms are clearly in very, very, very low concentrations. They are actually very difficult to find. So if you have antibodies as a treatment which are not targeted to the oligomeric forms, they would probably have very, very difficulties to find them. And I think also beside that, it can also become a cost issue, because we know that antibody treatment, we have seen that already in different therapeutic areas. They cost about $20,000-30,000 US dollars per year. I don't know if that's going to be the case with this one, but that's the area where we are playing with antibodies. And if we are going to treat patients preventive early, before we see any side effects or any symptoms, it's going to be a lot of people who need a lot of treatment for a lot of money. So I think it's a great value to use the body's own immune system, which is the path that we have taken, so that you can easily trigger the immune system to make the body itself produce the antibodies which are targeting the oligomers. And that's, I think, is maybe the only treatment which is possible to give for preventive treatment, because it's possible to treat on a broader base, not only for cost reasons, but also for practical reasons. I mean, you need to go every second week for a shot, and maybe that can make big influences for people. Whereas with an active vaccine, you're more free to come to the hospital maybe once in 6 months or once in 12 months. We don't really know yet, but that's the area where we are looking.
I just want to comment on that. I don't believe that antibodies for all the time will need to come to the hospital. I think that's a common misunderstanding. So I think there will be possibilities, I think, in the future for home infusions. And then I think we will see more and more development of subcutaneous administrations as well. So I think that will be different ways of treating antibodies as well.
And just to add to that, I think I want to echo obviously what everybody said, but I mean, we've been talking about primary prevention, we've been talking about secondary prevention, and I mean, these, again, talking about the long length of the disease progression and the huge amount of people, there will clearly be possibility for different types of therapies here. And when it comes to, certainly when it comes to secondary prevention, I think the small molecule compounds such as alstatin, our gamma secretase modulator, is highly suited for that role, kind of affecting the production of amyloid at an early stage of the disease. And actually, there are this really interesting Icelandic family, which, in contrast to the rest of the known mutations in the amyloid gene, actually has a mutation that prevents them from getting the Alzheimer's disease. And the mutation actually only causes a 20 to 30% reduction in amyloid production, and that prevents them from getting the disease. So I think, again, coming back to if we start early enough, then you can actually prevent the buildup of amyloid and actually the advent of the disease. So I think that's clearly important. Another thing that I think we haven't discussed much about is also symptomatics, because clearly there's a large population group of people that are in the later stage of the disease that also need effective therapies. So I think there's clearly a strong need for better symptomatic therapies, both when it comes to the cognitive symptoms, but also when it comes to the behavioral and psychological symptoms of the disease, which is a major problem for both the affected but also their relatives. So I think that's an important point to make.
I think we can agree that many challenges remain. There's, though, comfort in seeing that there are strong beliefs in your concepts, your various concepts, and we are looking forward to following the progression of your projects and on a global scale as well, of course. And hopefully we'll be here anytime in the future and talking about what was actually right, what was the solution. So we are running out of time, so I would like to thank you, all of you, for coming today and sharing your knowledge and your input. Thank you very much.
Thank you very much.
Thank you.
Tack så mycket.